Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. Today, as you can see, we want to navigate through the double helix structure of the DNA molecule as well as the DNA replication. And in previous lesson, that was lesson 4, we discussed the double helix as two parallel strands of a DNA twisting on one another to form the double helix. And then yesterday we discussed that the DNA molecule consists of three components. That was the carbon 5 carbon sugar, the phosphate molecule, and the nitrogenous bases. And under nitrogenous bases class, yesterday we discussed the four nitrogenous bases, that is base pairs, that is the adenine, guanine, cytosine, and thymine. And we also looked at yesterday about the base that pairs with one another. For instance, a base like adenine pairs with thymine simply because they were having same quantities. And therefore, a base like cytosine also pairs with another base called the guanine. And therefore, class, in a double helix structure of a DNA, what happens is the base pairs always, those that have same quantities, they pair with one another. That's adenine against thymine, guanine against cytosine. I just fill up one of the structures that I want us to fill together the others. For instance, let's say here we have the adenine, opposite we will be having thymine. Let's say here we have the guanine, here we will be having cytosine. And if we have here cytosine, here we will be having guanine. And for instance, we have here thymine, here we will be having adenine. And it is like that. The base pairs are found in a double helix structure. And we said a double helix structure are just basically two parallel strands of a DNA that twist on one another. And on the opposite sides of the double helix class, the bases are arranged, those that pair, for instance, the adenine against thymine, guanine, cytosine, and like that. For instance, let's continue. Let's say again you have adenine, it will be having thymine. Let's say you have base like adenine, thymine, cytosine, guanine, cytosine, thymine, and adenine. And for this is just a rough sketch, but not a diagrammatic exact double helix structure. But for the essence, is just to get the content and the basic knowledge in it, because it is well captured, a very good diagram in your KLB books. But the best thing is, are we able to comprehend whatever that is captured in that way? And therefore, this is just a rough sketch. Now we have also, again, let's say have the thymine. We will also be having here the adenine, guanine, cytosine, adenine, thymine, cytosine, guanine, adenine, thymine. And you can see, class, that these are the arrangements of the bases in a double helix structure of a DNA. This is the double helix structure of a DNA. Class, this is one strand, and this is also another strand. And we can see that these two strands are twisted on one another. And on the either side of each strand, they are the arrangement of the bases that are the nitrogenous bases, the four nitrogenous bases. That is the adenine pairing with thymine, cytosine pairing with guanine. Just these alphabetics just represent, the A represent the adenine, the T represents the thymine, the C represents the cytosine, and the G represents the guanine. Most importantly, class, today I want us to focus on the DNA replication. The DNA replication. The DNA replication. DNA replication. Most importantly, before we start looking at a DNA replication, the process of the DNA replication, it is very key, very critical, and very important. We need to understand what the term DNA replication means. DNA replication essentially is the fundamental process. Where the DNA is copied, the exact DNA is copied itself. That is what we mean by the DNA replication. And therefore we say DNA replication is the fundamental process by which a cell copies its DNA. It is the fundamental process by which a cell copies its DNA. Fundamental process by which the fundamental process by which a cell copies is DNA. And therefore DNA replication basically means the fundamental process by which a cell copies is DNA. Now, why is there importance in this replication? For instance, during cell division, and we know that cell division basically takes place in there are two types of cell division. You have the mitosis and the meiosis. Both of them involves division of the cell, but now the one among them results into two daughter cells, each having the same number of chromosomes as the parent cell, that is the mitosis, while the other one, meiosis, results into four daughter cells, each having half the number of chromosomes as the parent cell. But in each case, the genetic materials are just passed on exactly as the parent, because we use the parent DNA as the template under which the information is being copied from. And therefore, DNA is a very crucial during cell division. How? It ensures that the genetic information or the genetic material from the parent is exactly copied into the offspring. And therefore, that is why we say inherited characteristics are passed on. That is why this clearly explains, the DNA replication clearly explains that hereditary materials are inherited. How? Because the hereditary materials are contained in the deoxyribonucleic acid, that is the DNA. And what we know, the DNA in the parent is exactly copied to the offspring, the cell that makes up the offspring. And therefore, that means the genetic material in the parent now acts as a template under of which it is copied to the cell of the offspring. And therefore, the DNA is the ones that contains the hereditary materials. And as we said yesterday, that the DNA is like a long instructional manual that contains various codes and instructions for various traits and characteristics. And for instance, a particular trait or a character is contained in a gene, and therefore these genes are found on the long strands of a DNA which are contained in the chromosome. And therefore we know the chromosomes also consist of the chromatids, and along the length of the chromosomes we have the genes, and the genes make up the deoxyribonucleic acid. Now I want us to look at what happens during the process of DNA replication, or rather, how is DNA replicated, or rather, how is the DNA in the parent exactly copied to the cell of the offspring. That is what we want to look at today, and I want us to navigate, delve and dive through the process of DNA replication. Class, as you can see, we said that the double helix structure of a DNA consists of two parallel strands that are twisted on one another, forming long chains. That is the double helix structure. And as you can see, during the process of DNA replication, class, what happens is the double helix unwinds. We say the double helix unwinds and the two strands unzip themselves. During the DNA replication, the double helix unwinds, unwinds, and the two parallel strands unzips themselves, and the two parallel strands unzips themselves. Class, what does that mean? What we know, class, is these are the two parallel strands. Now the double helix is going to unwind, and these two parallel strands, they will unzip themselves, and as such we will be having the two per now, this one, they are, at the moment they are twisted. But during the DNA replication, this one, they will be separated. They will be in separate strands. That's what we mean, the unzipping themselves. And normally, we just use, when we zip, that means we close something, and when we unzip, we open. That means, in a way, this one, they will open up. That's what we mean. The two strands in the double helix structure of a DNA, they will open up and they will be in separate strands now. That's what we mean by the term. And zips from the term zipping. Zipping means just closing, and zipping means just opening. And in such a scenario now, the two strands in the double helix structure will unzip. That basically means that the two parallel strands will separate from one another. And therefore, each and every strand will be on their own. Now, what is the reason behind? So you can just state the reason, or what enables the DNA, the two parallel strands, which are twisted, to unzip during the DNA replication. Because class, you must understand again, what is the connection, what holds between the base pairs? That's a very important thing. What is the thing that holds the two base pairs? For instance, a base like adenine and a base like thymine, what holds? Guanine, cytosine, what holds them? Now, what we know, class, is that the base pairs are held together to one another by weak hydrogen bonds. The base pairs are connected together by weak hydrogen bonds. And as such, therefore, class, unzipping is therefore as a result of breaking, as a result of breaking of the weak hydrogen bonds that was holding the base pairs together. And therefore, this now sets the strands free from one another. Because, class, if the bond that was holding this to this is weakened and it has broken, that means there's the possibility of the two strands separating and opening up from one another now is very high. And therefore, the reason as to why the DNA, the double helix structure, unzips and unwinds is due to the presence of the weak hydrogen atoms. Now, once the weak hydrogen atoms are being broken, that means the two parallel strands will unzip, or rather will open up from one another, and therefore each strand will be free from the other, and therefore each strand will be free from the other. Now, if I do now a review of whatever I was saying, that we said the double helix unwinds and the two parallel strands unzips themselves during DNA replication. Now, we want to explain why is that happening. Why is the unzipping and unwinding of the double helix structure occurring? This is simply because we said that the DNA is basically made up of two parallel strands twisted together. And along the length, along those parallel strands, we have base pairs which are of similar quantities pairing on one another on the opposite sides of each strand. Now, what happens is these base pairs are held together by weak hydrogen atoms. Now, once these weak hydrogen atoms are broken, that means now the two parallel strands will be set free from, each strand will be set free from the other. Very good. Again, after unzipping, what happens? After unzipping, the information contained on each and every strand will be copied out onto a new DNA structure. After unzipping, the information contained on each and every DNA strand will be copied onto a new DNA structure, and the parent DNA will now be acting as the template to which this DNA is being copied, because the template from which the DNA coding form is copied from is the parent, which is acting as the template. Now, and therefore, the DNA is said to have replicated. And that is how the process of DNA replication occurs, class. Another important thing, class, is I want us to know how does a replicated DNA looks like. Now, if I now summarize this, what I mean, class, is that the DNA replication is the fundamental process by which a cell copies its DNA. Very good. Again, during the DNA replication, what happens is this double helix unwinds and the two parallel strands unzips. The reason also why the two parallel strands unzips is because the base pairs are held together by weak hydrogen atoms. And once these weak hydrogen atoms is being broken, that means the two parallel strands will set free from the other, and therefore the information again on each and every separate strand will be copied onto the new cell. For now, the parent cell acting as the template onto which the DNA is being copied from. And that is why, and that is therefore the way DNA is being replicated, class. Now, class, how does the DNA, replicated DNA looks like? For instance, this was the one that was there, for instance, and again we have the DNA, right? The parent DNA. Again, let's look at the replicated one. The replicated DNA will be similar to the parent DNA class. For instance, let's look at this. This one will be adenine, thymine. You see now, they are just similar. That's why we say the DNA will be replicated the way it is exactly. This one is guanine, cytosine. Again, adenine, thymine. Are you seeing adenine, thymine like that? Thymine, adenine, exactly the way it is here. Cytosine, guanine, exactly the way it is. Guanine, cytosine, exactly the way it is. Adenine and thymine. That is a replicated DNA. Why? Because the replicated DNA, they are similar. There's no difference. The arrangement, the coding, everything looks similar, and that is why we say the DNA is copied exactly the way it is. Now, class, if I want to look at the roles of a DNA, what are some of the roles of a DNA? Roles of DNA. Class, role number one of a DNA is stores genetic information in a coded form. It stores genetic information in a coded form in the nucleotide basis. We know in the nucleotide basis, it stores genetic information in a coded form. Number two, it transfers the genetic information to the daughter cells. It transfers to the daughter cells unchanged. Yani, that means it simply means that the genetic information being passed from the parents to the daughter cells is similar to that of the parent without being unchanged, and that is being ensured by the deoxyribonucleic acid. Now, lastly, genetic information, DNA translates genetic information into characteristics. Translates genetic information into characteristics. It translates genetic information into characteristics. Through what? How can a DNA translate genetic information into characteristics? Simply by doing through protein synthesis. Through protein synthesis, or through protein manufacturing. Through protein synthesis. Very good. Now, those are the three roles of DNA. Roles of a DNA. It stores genetic information in a coded form. It transfers again genetic information. Now, can be asked, how does a DNA stores genetic information? It stores in coded form. Where does it store again? In the nucleotide basis, right? It transfers. How can we know that the DNA from the parent is exactly the one in the offspring or in the daughter cells? That is being ensured by the DNA, such that the DNA from the parent is transmitted to the daughter cells and changed, and that is being ensured by the DNA. And lastly, the DNA translates genetic information into characteristics. How can the DNA translate genetic information into characteristics, class? through protein synthesis. Can be a very simple question. Explain how the following, how does the DNA ensures the following. Number one, that the genetic information remains unchanged, or the genetic, how does the DNA translate genetic information into characteristics. Very simple questions like that. And that is briefly what I wanted us to look today. And in our next class, we will be looking something different. But for today, I will wish to stop at that point and will wish you all the best success in your future endeavors. And may Allah subhanahu wa ta'ala guide us through the straight path. اهدنا الصراط المستقيم